For this lecture, we will discuss molecular diagnostics and other calculations that we have not previously covered. The clinical molecular laboratory has become an integral part of the diagnostic testing menu. Actual viral loads for the human immunodeficiency virus, or HIV, or even hepatitis B or C has dramatically changed treatment options for these patients. It's no longer good enough to just be able to measure the presence of the actual HIV antibody in these patients, but treatment protocols now demand that we know the exact amount of virus that is present within the body. Now, with recent events of older viruses kind of emerging in a newer way or a newer form, this has kind of showcased how vital the role of molecular biology is for laboratory professionals, so that we can respond to these new changes in viruses with new analytical techniques to kind of aid with the diagnostics. Now, we have covered in previous lectures the metric system. However, in molecular biology, you need to understand that much smaller quantities of materials and dilutions are actually used versus what we typically would do in a core laboratory or within a chemistry or hematology laboratory. So, for example, if a 1 to 10 dilution was going to be performed in clinical chemistry, then that dilution might be performed by actually taking a 10 microliter sample and mixing it with a 90 microliters of a diluent. Whereas in your molecular biology testing, you would use a smaller quantity to make that 1 to 10 dilution. So you might use something like 1 microliter of sample to 9 microliters of diluent. So you would actually reduce that quantity that you would use in chemistry, you would reduce it to an even smaller quantity for molecular biology. So where your total volume in your chemistry dilution would be 100 microliters, your molecular diagnostic solution would actually be 10 microliters. So it would actually be 1/10 of what you would use for your chemistry testing. So here we have a nice little chart just to kind of remind us again some of that terminology that we use within the metric system. We've already talked a lot about things like the deci, so like deciliters, milliliters we've referred to, microliters. So you've already kind of seen some of this terminology. So obviously, as we're kind of moving over to the molecular standpoint and we're going to use some smaller measurements, you're going to start seeing some of these other terms being referred to as well. So buffers are actually used in the clinical molecular laboratory for a variety of tests. So for example, if a sample is actually tested by electrophoresis, then those buffers are actually an integral part of that electrophoresis technique. So common buffers that are actually used are the triborate buffer, the TE buffer, the TAE buffer, otherwise known as triacetate EDTA, the STET buffer, and the maleic acid buffer. So there are calculations that we've used in the past that you are probably still kind of familiar with, is getting those concentrations based off of those volumes. And so we can still use that when we're working with a buffer from a stock solution, as far as being able to make those concentrations of things that we need. So the difference between an actual, your main laboratory or your chemistry department where you're going to do those electrophoresis procedures or other procedures that require a buffer, the difference between the two departments will actually be between the volumes that are actually used. So because of, in the molecular laboratory departments, you're going to use a smaller quantity of the patient sample and the patient's reagent, because we're looking at stuff at a molecular level, so we don't need as much volume to actually do that testing. Whereas when you're dealing with something like in the main laboratory, you're going to have larger volumes and you're going to need, kits and things with larger containing volumes of pre-made reagents that you're actually going to use to make those solutions that you need. Now, with some of those various techniques that are actually used in molecular biology, like for example some of those extracting techniques, one of the common things that is going to be used will be ethanol and a lot of times it has a 70% volume per volume concentration. So let's look at an example of where we might actually start using some of this math in the actual molecular department. So in our example here, a medical laboratory scientist student is preparing a 70% volume of ethanol solution to actually be used in a hepatitis C molecular assay. So what we want to do is actually use the stock concentration and the volume that we have listed here to actually determine how much we will need to actually prepare this particular solution. So if our current stock solution of our ethanol is at 100% volume per volume, and we need to make 100 mLs of a 70% solution, then we need to know exactly how much we will need of each. So again, we're going back to using that calculation of where we have a concentration of our stock solution and a volume of that stock solution. We need to know how much of that stock solution do we need. So currently we know that our concentration is at 100%. We don't know how much volume we will need, but we also know that we're trying to reach a concentration of 70% that is at 100 ml volume. So once we plug all of those numbers in to where they need to fit in that equation, and we can start solving for that, then we'd use our algebraic math, and then we come to a determination of 70 mLs is what we will need of our stock solution to be added to a 100 ml flask in order to make that dilution with deionized water. So 70 mLs of it will actually be our ethanol, and then 30 mLs will be of deionized water will be added to that, and that's going to give our 70% volume per volume solution. Now, as we proceed to some of your future courses, we'll go into a little bit more detail of the actual molecular biology lab and what actually is done in there. But just for now, just kind of understand that our DNA, whether it's going to be referred to in terms of our double-stranded DNA or our single-stranded DNA, or even down to the oligonucleotides that are actually extracted when we're doing this testing, that their quantity may be expressed in molar terms. So we actually have a chart here that kind of lists how they are expressed. And so it kind of lets you know that the molecular weights of DNA, RNA, and oligonucleotides are actually reported out in terms of picograms per picomole. So in the molecular laboratory, you might have to use some of these conversions. So there are a few formulas that we can use to do some conversions. One of our first ones is going to be to convert double-stranded DNA from micrograms per ml to the picomoles per microliter. And so that will use the following formula, where you have your X as your unknown number of microorganisms per ml of DNA. And so what we're doing is we're looking to see how many of those organisms are we detecting. So we would plug in to that equation would be our X number of micrograms of our double-stranded DNA times the picomoles per 660 picograms. So that's that actual molecular weight for that double-stranded DNA. And then we're going to multiply that by 1 ml per 1,000 microliter, because we know that there's 1 ml in 1,000 microliters, times 10 to the 6 picograms per 1 microgram. And this is all just involved in doing that conversion, times 1 divided by the number of nucleotides. And so that would be your equation to figure that out. There's also an equation to convert from the picomoles per microliter of DNA to the micrograms per ml of DNA. That conversion's here again using that standard molecular weight. And then if we were going to do these conversions with single-stranded DNA or RNA, then we would use that 330 picograms per picomole in our conversion formulas for that particular molecular weight. So here we have an example where we would actually use some of these formulas. So a new employee was told to convert 12 micrograms per ml of a concentration of a double-stranded DNA sample that contains 2,500 base pairs into units of picomoles per microliter concentration. So we want to know how much DNA in picomoles per microliter does this new employee have in this particular sample. So we would substitute those values out and apply it to the formula. And again, we're using that double-stranded DNA, so because of that, we're going to use the 660, the picomoles per 660 for that molecular weight. Plug that all into that formula, and when we do that math, and that is going to give us a final number of 7.27 times 10 to the negative 3 picomoles per microliter. Now, sometimes the amount of DNA that has been produced and is quantified, so you can actually get a number value to it via the spectrophotometer using the turbometric method. So when a substance is turbid, it will actually block the light path of that spectrophotometer. So if it's got particles in there, it's turbid, it's not clear, then it's obviously going to block some light. And so this is going to lead to a decreased transmittance of light that is coming in contact with the actual photodetector in that spectrophotometer. So this decreased transmittance is actually a measurable component of spectrophotometry, and so we can actually measure it at two different wavelengths. We can measure it at 260 and 280 nanometers in order to do DNA analysis. So just an example of how this is used in research laboratories, they can actually take this measurement in order to quantify bacterial growth in some cultures. In the actual clinical molecular laboratory, the ratio of this measurement of the optical density of 260 versus 240 should be between 1.8 and 2.0, and it will actually indicate the purity of the DNA that was actually extracted from that sample. Values greater than this are an indication of a contamination of that DNA. So with that being said, there are actually conversion formulas that have been developed that will actually convert the optical density at 260 nanometers to micrograms per ml quantity of DNA or RNA. So by using ratios and proportions, the micrograms per ml quantity of DNA or RNA can actually be calculated once we know the optical density at 260 nanometers, and we've determined what that is. And here you can see a table that actually lists the relationship between that optical density at 260 nanometers and the amount of microorganisms that are contained per ml, and that's based off of if it's reading at an optical density at the 260 nanometers, the equivalent of how much would actually be tamed is based off of that particular DNA, whether it's double-stranded, single-stranded, or RNA that's present in that sample. So let's go over an example of how we will actually apply that to the actual molecular laboratory. So in this example, a factor II assay has been performed. One step was to actually extract the double-stranded DNA and determine the amount of micrograms per ml quantity so that further calculations can actually be performed. So the optical density at 260 nanometers was measured, and we had a value of 1.425. So we want to know, what is the micrograms per ml quantity of that particular double-stranded DNA in that sample? So what we'll use is use some of this previous knowledge in the previous tables to actually determine what that's going to be. So we kind of got to look at it from a ratio proportion standpoint. So if we know that one optical density is equivalent to 50 micrograms of double-stranded DNA when read at 260 nanometers, then we can plug in that when we read our unknown sample and we get an optical density of 1.425, then we'll plug that in so that we can help to determine exactly what that's going to be equivalent to in the amount of microorganisms per ml. So when we kind of plug those numbers in and work out that equation, then we'll come to a final figure of 71 micrograms per ml. So we know that when we read that optical density for that double-stranded DNA for that particular organism that's in that sample, then we can know that we have about 71 micrograms of that double-stranded DNA per ml in that particular sample that we just did our testing on. Now there is another type of solution that you should be familiar with, and it's one that consists of a hydrated chemical salt that is actually dissolved in a solvent. So there is actually a difference between the hydrogous and anhydrogous form of a chemical, and it has to do with the amount of water molecules that are actually present in a hydrated form. So because of this, it may be necessarily that you may actually have to interchange between the two. So sometimes in the laboratory, only one form of the chemical is actually available, so you must be able to actually interchange between the two forms in order to come up with the form that you're actually going to need. So there is a basic formula that's listed here that allows for the interchange in between hydrated and anhydrous chemicals. You'll notice that it is a ratio and proportions calculation. So a ratio and proportion is actually used whenever the same concentration of a solution, you have the same concentration of a solution, but you have different quantities of that solution that may be necessary. So in an anhydrous, as opposed to a hydrated form of chemical, then that ratio and proportion is actually used to ensure that the same concentration of the chemical is being used regardless of how much water content is actually in it. So now let's look at an example. So suppose you need to make a 12% weight per volume solution of copper sulfate, but the only thing you have on hand is some copper sulfate solution and water. So we got to figure out how much of that copper sulfate solution do you need to mix with how much water in order to obtain that 12% weight per volume. So we need to first determine our molecular weights. We need to know the molecular weight of the copper sulfate individually, as well as mixed with the water of what that molecular weight would be. And then from there, what we need to know and determine is how many grams are actually in a 12% weight per volume solution. So we know that if we have 12 grams in 100 mLs, that's going to give us a 12% solution. From there, we use our formula for interchanging the hydrogous and anhydrous chemicals and plug in all of those values. So we plug in what our known molecular weights are for that, and we're trying to figure out exactly how much of that copper sulfate do we need. So we plug in what we'd expect from 12 grams of copper sulfate in a 100 ml solution. And then we work out that math, and then we come to a final conclusion of 13.4 grams of copper sulfate is what we would place into our mixing container and add that water to reach that 100 ml volume. And then that's going to give us a 12% weight per volume solution of copper sulfate. So there are actually some acids and bases that may have the specific gravity actually listed on that chemical label. And so this is important when the mass concentration per ml of acid or base must be known. So the specific gravity is the ratio of the density of the chemicals in terms of the grams per milliliter compared to pure water when we actually do this at 4 degrees Celsius. So that's about refrigerator temperature. So pure water has a density of 1.000 at 4 degrees Celsius, or 1.000 grams per 1 ml of pure water. Therefore, we know that using pure water, there is a direct relationship between mass and volume. So based off this, we know that the specific gravity of a solution is actually equal to the density of that solution, and this is going to be in terms of grams per milliliter per solution. So now let's talk through an example. We have a 1.00 liter bottle of sulfuric acid, and it is labeled as a 92% weight per weight with a specific gravity of 1.84. So we want to know how many grams of that sulfuric acid is found per ml. So first we need to determine the concentration of the sulfuric acids per ml. We know that if the solution has a 92% weight per weight, then there are 92 grams in 100 grams of solution. So therefore, using the ratio and proportion, we can determine the amount of grams of that sulfuric acid in 1 gram of our solution. We plug those values in, cross multiply, and then that gives us a value of 0.920 or 0.920 grams of sulfuric acid in 1 gram of solution. But we're not done there because we want to know the actual amount of grams of that sulfuric acid per ml of solution. So the specific gravity of that sulfuric acid solution is 1.84, which means there are 1.84 grams of sulfuric acid per 1 ml of solution. But only 92% of that 1.84 grams is actually that sulfuric acid. So then we need to do an additional formula to determine the amount of grams of that sulfuric acid in that 1 ml solution. So we'll plug those values in here into this equation for what we already know. And then once we do those calculations, we'll come to a conclusion that there are 1.69 grams of sulfuric acid per ml of solution in this particular bottle. So a blood gas analysis is going to contain several things within that laboratory results. You'll have your pH, your partial pressure of oxygen, your partial pressure of the carbon dioxide, your bicarbonate, your base excess, as well as your percent oxygen saturation. So the first three are measured by the blood gas analyzer, whereas the last three are actual calculations. And the percent oxygen saturation, which is otherwise is listed as a percent SO2, it can actually be determined by a pulse oximeter or estimated from a nomogram. Now the percent oxygen saturation, yes, that particular calculation is actually dependent on how much hemoglobin is available, so that hemoglobin concentration, and then its ability to actually bind those oxygen molecules. So each molecule of hemoglobin is capable of binding 4 molecules of oxygen if a person is actually healthy. And so that means that his or her CO2 will actually, his or her percent of the oxygen saturation is actually going to range anywhere from 96 to 98%. So if you have a percent oxygen saturation that's above 100%, which is almost impossible for someone who is breathing in room air, but a situation like that is most likely going to just be found in patients who have some sort of respirator. So the percent of oxygen saturation is rarely calculated by the laboratory professionals, but if you do need to do a calculation for whatever reason, then that calculation is listed here. So to do that, you actually have that percent saturation over that CO2 per hemoglobin. That CO2 is the concentration of the oxyhemoglobin. And then you'll put that over the concentration of the deoxyhemoglobin. So again, the way the instrumentation is now, it's not likely that you'll need to do this, but there are still some registry questions that want to know what that calculation is based off of. So another calculation that you need to be familiar with, we really don't have to do it a lot anymore because the analyzers, you can plug a lot of this stuff in and the analyzers will do these calculations for you, or your LIS. But just so that you're familiar with how to do them, just for registry question purposes, We will kind of talk about these a little bit. So the chemistry tests performed during a urine analysis are actually qualitative in nature. So in other words, we're getting more of a positive, negative, or a range area based off that visual reaction that we see take place on the strip. So, they can indicate that there's a problem if they're abnormal, but sometimes you might need to follow up on those to do some additional testing. So a physician could potentially request a 24-hour urine to actually get a quantitative analysis of whatever we're looking for in that urine sample. And so that quantitative analysis, again, you got that N in there, so that's going to give you an actual number versus qualitative where you get a letter, a positive or negative, or kind of a range area. So let's just say, for example, a 24-hour urine collection is performed and they're looking for a total protein analysis or let's just say they're looking for urea or anything like that. They're trying to see how much of that is actually being cleared from the system in that 24-hour period. So as you would expect, when you're collecting that whole total volume for that 24-hour period, then when you test it, that you're not testing that whole quantity. You're going to pour off a little bit and you're going to test that little bit of sample. So what you're going to have to actually do is do some calculations where you figure out, well, if this is what's in this small volume, then what's in this larger volume. So you kind of got to use some of those other data that we have to actually apply it to some mathematical calculations. So because the volume and the duration of the calculation of the urine can be variable, then those results need to be standardized to allow to be able to compare results. Now, most of these results are recorded as a quantity of an analyte per a unit of time, and so usually that time is going to be 24 hours because that's about the typical range that we would do for a 24-hour urine collection. So occasionally a urine sample will be collected for a shorter period, whatever the case may be. So in those cases, the quantitative urine results can be reported as a quantity per volume of collection or a quantity per unit of time. So this might be something like a random collection, so that you just test it off of just a little bit of a sample versus that 24-hour period. So now that we've kind of briefly covered why we're doing the testing, let's go a little bit deeper into what it's actually going to involve. So let's just say, for example, in general, a non-electrolyte is measured in the urine, and then we're going to report that in units of milligrams per day or in our international units of millimoles per day. So the urine electrolytes are generally reported in units of milliequivalents per day or millimoles per day. But if urine is not collected for that whole 24-hour period or that one-day period, then the results will need to be extrapolated for 24-hour collection for compounds that are uniformly excreted during that 24-hour period. And of course, they'll have to be reported in terms of per volume quantity collected, or that quantity per that total amount of time that was actually used to collect that sample. So in order to actually calculate the quantity of that volume of collection, then we're going to have to use a ratio and proportion calculation again in order to figure out those values. So here we have an example. We have a urine specimen that was collected for 12 hours, and the order for it is to do a creatinine analysis. So our volume over that 12-hour period we know is 800 mLs. The creatinine result, when we run it off of an aliquot of that sample, it's going to give us 90 milligrams per deciliter. So what is that creatinine result in milligrams per the volume of that collection? So to solve this problem, we're going to use that ratio proportion equation to calculate it out. We've got to remember that we've got to make sure that our units are the same. So in order to do that, anything that needs converting will need to be converted. So in this particular equation, we'll have to convert that deciliter into milliliters. So that means that 1 deciliter is 100 milliliters. So again, we figure out those volumes, plug all of those in, work that equation. And so for this particular example, for this patient, over that 12-hour period, then we have determined that there are 720 milligrams of creatinine per 800 mLs of urine. Now within that same example, let's just say that we want to know the amount of that particular analyte that's collected in that urine per unit of time. So we want to figure out its proportion in relation to time. So the calculation is pretty identical to the previous calculation, with the exception of reporting it as milligrams per collection period, versus whereas we figured it out per milligrams per that whole entire volume. So unless a time other than 24 hours is specified, then the unit time would be 24 hours. But let's look at the previous example where we talked about it being collected over 12 hours. So we already know that the creatinine in 800 mLs that was collected in that 12-hour period was 720 milligrams per 800 mLs. So because of that, then we can further calculate that that basically we can just report that out, that 720 milligrams was recorded and cleared over that 12-hour period. And so again, when we're kind of talking about urines, you're kind of already seeing that we can kind of look at it over how much is cleared per particular volume versus how much is cleared per particular time frame. So there's a couple different equations and ways that we can apply that. Now, additionally, we can kind of look at the quantity of an analyte to figure out how much of that concentration is going to be in there in grams per day. So there's some conversions that can do that as well. And an example of this would be a urine urea value of 60 milligrams per deciliter is obtained off of a 24-hour urine. And we have the volume of that whole volume collected in 24 hours. So this is how much this patient was able to clear volume-wise over a 24-hour period. And then we know how much of that concentration of it is actually urea within that 24-hour period. So we can kind of gauge a few other things based off of that. So if we do some substitutions and plug all of these things into the conversion formula and we work out that formula, then we can further gather from this that the patient's urea concentration is about 0.9 grams per day, or 0.9 grams per a whole 24-hour period that they're actually clearing from that. So a lot of this will be used to kind of gauge various states and health of the kidney or the urinary health or the overall health of the patient in general. So you'll learn more about those as you get a little bit deeper in some of your other courses. But for now, we're just kind of looking at the math aspect of it. Again, a lot of this stuff, you're just going to plug the values into your LIS or your computer system for your analyzer, and it's going to do all these calculations for you. So one of the things that I think that some textbooks are not really mentioning a lot of now, but I know this is still probably going to be a good registry question, and I think it's one of the things I failed to neglect to put in the hematology lecture, but you still do need to know this, and that is to actually be able to get an absolute count when doing that white blood cell counts and differentials. So our total count is actually going to be our total number of white blood cells that are in circulation. So when we do that peripheral blood, run that blood through the analyzer, and we get that count of white blood cells. This is what we're talking about when we talk about a total leukocyte count. Now, our relative count is that percentage. So when we do that differential, when we divide up those cell populations between neutrophils, lymphocytes, monocytes, etc., that is our relative count. That is our percentage. Then we also have something called an absolute count. And so what this is going to do is actually provide a little bit more of an accurate measurement of the actual number and types of cells that are present in that peripheral blood smear. So there is an equation that will do that. And with that equation, we use the relative count divided by 100, and then we multiply that by the total white blood cell count. So the situations where this might happen will be when you run a CBC through your analyzer and it gives you a white blood cell count, but for whatever reason, there's some things that are interfering. So it's not getting an actual very good differential count. So it might block out that percentage and that absolute value in your printout. That means you're going to have to actually do that manual diff. So for whatever reason, if something's interfering, if there's some immature cells or some nucleated cells, platelet clumps, anything like that that could be interfering, then you switch over, you make that manual slide, you do that diff count, and you get that percentage. And from there, you can actually calculate those absolute values to determine, based off of that population or based off of that patient's cell count. So if their cell count's really high or really low, it can kind of skew those percentage numbers a little bit on you, especially when you're doing those diffs and you're only looking at a certain population of that slide, not the whole entire slide. So this kind of helps give you a gauge for those absolute numbers. So here we have an example where the patient's white blood cell count ran on the machine was 7.0, and then you do your manual diff and you get a 70% count. So we want to figure out, based off of the absolute count, is that high? Is that kind of normal? What is that? So we'll be able to kind of put it in terms where we can actually look at it through those absolute reference ranges. So if we plug all those values in, then we get an absolute count of 4.9 times 10 to the 9th per liter. And then this just kind of helps us to assess this when we weren't able to get those absolute values off of that printout.